Class, what do you think of that? All right, here we go. So, this is this week. Hey, we had another day last Thursday, so we're catching up with that. Remember that recording I made in two parts is as important as a class meeting, and you got to watch that now if you haven't already watched it. So, what should you do this week? So, you're going to finish reading chapter 16. Chapter 16 is about receptors, and is probably the most significant chapter in our book. And so because of that, and that it's hard to understand, we're going to do a homework assignment, and it's based on a video that I'll add a link to in Brightspace today. And what you're going to do is learn the whole video. It's actually a really long video. It's like 45 minutes long, I think. And it's about what happens under a scab to get your skin cells to heal. So that's going to be our focus of an example of a receptor signaling pathway, because it tells us about the most important class of receptors in chapter 16. It gives us a chance to already start leaning into chapter 17, which is about the cell cycle. And under a scab, the skin cell cycle gets stimulated by this growth factor through this receptor signaling pathway. And then 3, there are 2 famous oncogenes, cancer-causing genes, that we'll learn about. One is RAS and the other is MYC. And the author of our textbook first learned about RAS in Wisconsin, and studying retroviruses, and then he left the retroviruses behind and studied the animals. And so that's what we're doing too. We'll bring up those oncogenic retroviruses once in a while in our course, but really move past those because it's a host gene, it's an animal gene, and there are orthologs of RAS in all kinds of animals, so like that. So in addition to that, because it's a really important video, we're gonna have a quiz on the entire video at the end of class a week from today. And it's going to be short answer questions, similar to the quizzes we had at the start of our course, because this is another pivotal piece of learning example. So we learned about muscle differentiation in our course really to focus in on genes and regulation of gene expression. And then a universal definition of a gene and how genes function. And this is similar here too. We're just going to focus on one receptor, have a really good example of something we can take all the way through. And then we will learn about others too, lots of others. So we're going to have an assignment and a quiz based on the same video. So you're definitely going to watch that video multiple times. It's just crammed full of stuff that is the kind of the heart of our course, okay? And then get ready to start working on your term paper. So I'll probably before Thursday, because your term paper is going to be about this chapter. Each of you is going to pick a receptor and write a term paper about it. And the way we're going to do it is I'm going to share 2 Google Docs with you that you're going to work on. And so I mentioned this today because before Thursday, I'll probably share those with you, and you'll have questions. You'll be like, what's that about? You know, when I just share them, I'm just there, it's shared. And I'll explain on Thursday how those fit into our term paper assignment that's due, toward the end of the semester. All right, so I'm just going to kind of finish up some review of chapter 7, because chapter 7 was all about transcription regulation, and we drove through it in our class by picking up examples and focusing on examples to create a framework that you can then build out more on. And so the first one is that there are these 3 major ways to regulate transcription. And there are other ways, but these are the main categories. And we have nice examples that have specific transcription factors. In each of these categories, there are other transcription factors that work that same way. So there are other transcription factors that recruit TBP. They work just like MyoD does. Then there are additional other transcription factors that recruit other components of the pre-initiation complex, other proteins that are the rate limiting step. In some cases, it's just the RNA polymerase that needs to get dragged there to get it done. So that's that one. Then remodeled chromatin, and we have a really good example of that. And let me just scroll forward to show you this. So you don't have to read this in detail, but one time I taught this course, I taught students a little bit about this concept of mind mapping. And so this looks really messy, but mind mapping or concept mapping involves making a central concept like their estrogen receptor and then growing out in different directions with different concepts and topics. And we're not doing that this semester, but I wanted to show this because we've learned a lot of different things about the estrogen receptor. And it goes off in different directions, right? So, it's got different domains. We learned about that idea. It's a transcription factor that is regulated by a hormone. We learned about that. It's got a transacting domain that's really interesting because it can function as a repressor or an activator. And it's a member of the freaking nuclear receptor superfamily. And I haven't mentioned that word superfamily, I just say family. But when there's a group of paralogs, gene family, that has 48 members or several members, and they kind of are big, cover a big bunch of territory, people call that a superfamily. And so they're a superfamily, go out this way for the ligand binding domain. Oh, and there's tamoxifen, right? We learned about that in cancer, right? So see, that's a thing, right, where we just have tons of knowledge on estrogen receptor that we just picked up, right? And I don't know if they have this, but there's a method that, I'll just in review, that I talked about in the recording last week called CHIP that could be on here, chromatin immunoprecipitation, right? Coming off the DNA binding domain. That's how you identify all the estrogen receptor target genes, not just vitellogenin. Vitellogenin is probably on here, but other target genes. So I don't know, let's just out of curiosity, raise your hand if you've ever learned something about how to do this mind mapping or concept mapping. Have you ever done that in class? Like that? And so it's a powerful learning technique because, you see that we learned it, it comes up again and again, and we go out this way and go out that way. And it's all about estrogen receptor. And sometimes it's a lot easier to remember that, especially if you draw it big, than if you just have an outline, right, with estrogen receptor. It just doesn't come up that well in your mind as it does this, like that. And that's what people, the teachers who are really into concept maps, they're really into that fact that students learn material better if they draw it out like that. And you can Google that, and because, again, we've done this in an assignment in our course before, and I didn't really teach it. I think I had a link with some introduction to it, and I'm just like, go for it. But, it's a cool thing. The only issue here is to just make it big. Actually have it be, out in the woods, and you go from this area of the woods to that area of woods as you're explaining this different stuff, and then you go back to the, the original granite boulder that is the estrogen receptor. Yeah, it's a good thing. And I think I'm sure I mentioned that when I ran into this stuff in college, I just wanted to open my head and just tip it in, just tip that knowledge in, because I liked it so much. Yeah. And, so how do you tip it in and just have it in? And that's hard to do, but you're learning about that kind of on your own, but also part of our class. Okay, so then we learned about the estrogen receptor, and then we learned about this heat shock transcription factor that provides a nice third example. And its example is to release this arrested polymerase. It's a really sweet example. All those, of course, I picked in context to be, that's a memorable example. Heat shock transcription factor, there are orthologs of it in every eukaryotic cell on our planet. It's just a big deal for life on our planet. And it's like a first aid emergency thing when there's some kind of shock to a cell that causes a lot of protein denaturation. And the proteins are denatured, and they gotta be, working for the cell to survive. And so this got to scramble and try to refold those proteins. You really don't have time to just make them all new, you just gotta go, we gotta get out there and save some proteins, refold some proteins, and save this little cell's life. Okay, so that's just good. And see, it's a core set of stuff to navigate us through big chunks of the chapter. And this is just something I drew at some point, and I really encourage you to draw it out. In some cases, we have drawing assignments, right? But just across the board in this course, it's all physical. You just draw all of it out. That's like a concept map that's more literal. right? And so you could potentially draw that gene and then just start making concept map lines out from it. And you draw it big, and again, why do you draw it big? A world famous scientist told me this. He said, you know, Dave, the bigger you make it, the more you can learn, right? Because you just spread it out and you can put more on it, but you're also, you move, your mind moves as you recall that. So that's what we're trying to do, is we're trying to learn a bunch, and one of the big concepts is the rate limiting step. There's going to be a rate limiting step. We talked about that, okay? So then, it should be another one. Oh yeah, we have this list we're working on. We added that to this list, right? We got HSTF. HSTF is a really oddball. Very few proteins trimerize, they do, but there are a lot of transcription factors that dimerize. So the idea of needing to get together with another monomer is really common. So that's a good example for us on that list. And then there's this one, and that is that one of the big things the book wants to explain in chapter 7 is remodeling chromatin, changing chromatin organization, because the DNA is always folded up. And so we have a really good example of that in the estrogen receptor. I thought I had moved to that slide. Maybe I don't even have it. Yeah, there it is. That's what I was trying to get to. So we said, you know, we have that example with the estrogen receptor, and it fits in here. And then we have a little example, just a little bit of DNA methylation stuff. And then we have this one down here with the GAGA factor that fits into the heat shock transcription factor story. And that's a good example of another type of chromatin remodeling. So the three types of chromatin remodeling that we'd especially emphasize are DNA methylation, histone acetylation, and nucleosome phasing. And we have an example of each of those. Yeah. And so, you know, on the test, it's like short answer questions. And I'm like, give an example of, you know, chromatin remodeling, or give 3 examples of it. You'd be like, DNA methylation could be a list like that. Or I could say, explain what we learned about, you know, histone acetylation. Yep. Okay. So I talked about all that stuff, and then I covered this. And this is in chapter 7, and I think almost all those methods are in there. And so it's toward the end. Like I made a 2-part video last Thursday because I had to get a kind of drink of water, and the second part is only about 20 minutes long, and it's basically those. And it's methods that are in there. Matt was just talking about the method, and each one, it could be, you know, that could be 2-point questions. You know, like how would you do that? And what's that? You know. And we got to learn some of the methods in our book because they're in there, and they're in there to help, basically, to help illustrate the physicality of this research. You know, like, how'd you learn that? The answer is like, well, you know, we got to do a lot of experiments, and these are examples of that. And it's a nice list because it actually can be thought of as being linear. Like this would be done, these experiments would be done in this order. You start up going, I don't even know anything about how estrogen works, you know? And so you find a gene that gets activated by estrogen called the vitellogenin gene. And you're like, oh, well, how does it work? And the answer is it must be some part of the cis regulatory region of DNA. So let's do DNA deletion studies. Let's start chopping portions off until we have broken the cis regulatory region. And when that one is broken, we've mapped the location of that estrogen receptor binding site, that enhancer, the estrogen response element. And then we can do a nucleus footprint to show, yeah, there's a protein that binds to that. So we get a little footprint, and that gives us a piece of DNA that would be the enhancer. Then we can confirm that with this gel mobility shift assay. Yep, there's a protein that binds to that DNA, and it causes it to migrate more slowly on the gel. So then we can take that piece of DNA and stick many, many cups of that DNA onto beads and purify the receptor. Now we have the receptor purified, and we can purify enough receptor that we can make an antibody to it, and we use that to do the CHIP method. Chromatin immunoprecipitation requires an antibody to pull down the estrogen receptor as an example of a transcription factor, and all of the target genes. So not just the original vitellogenin gene we started with, but all of them. And I don't think I quite, and just thinking back right now, I don't think I quite got into the statement at the end of the video. I think I skipped over one slide, which was that the heat shock transcription factor is, it's got a job. and it says what its job is. It's got the heat shock response, this shock response, and it's going to activate several heat shock genes. And so people study that for years and years and years. And then here's just an addition to the concept of CHIP. And that is, when they did CHIP on the heat shock transcription factor, they pulled down all the heat shock genes. When the heat shock response is happening, it's activating. It's bound to all those. And they also found other genes that the heat shock transcription factor activates. And so they learned that the heat shock transcription factor actually plays a role in a bunch of different responses, not just that heat stress type response. So CHIP is super valuable to really find out, well, what is the full role of this transcription factor? We would love to think about doing CHIP with MyoD. And if we pulled down MyoD, we would pull down myogenin, but we'd pull down some other target genes that we haven't learned about, that scientists learn about, that are important in muscle development also, and maybe even some other stuff too, in other settings. So the CHIP is a powerful assay for transcription factors and identifying targeting. So I want to just kind of blab through that because I really talked about all that already, and we're really done with chapter 7, so I should pause because we had a snow day, and give folks a chance to ask questions from the book, and anywhere. You could be in chapter 16 already and asking questions from it, because that's where we're heading now, or anywhere else. Yeah, and one thing that's nice about this book is we can actually jump from chapter to chapter like this and not miss out. The book is building up stuff, but it's actually cleverly written where we can jump to chapter 16 now. And you don't have to look back at, chapter 12 or 14. It's kind of self-contained, yeah? And, but yeah, do people have questions about anything at all? Okay. Just always remember that time when I taught this course and I didn't say anything except answer questions. And Julian and another student were the only people that ever really asked questions, and that was fine. They asked a lot, But yeah, questions are a good thing, right? And so write them down. Like if there's a snow day lecture and you're looking at it, and you're like, I don't know, I would have kind of liked a further explanation of that. Yep. Also, when you go home and you should go home and look at these again, right? Because something you may have noticed is I talk fast because I'm recording it, right? I used to talk slowly because I was trying to make sure everybody got it because it was just going away in space, right? But it's not true. Now you can go home and slow it down, right? Literally, pause it, stuff like that. So, if you do that and you go home and you say, well, I still don't get that. How could you? I mean, it's a lot of new stuff, So, yeah, ask a lot of questions. So this is chapter 16. And chapter 16, and you don't have to scramble to write this down, but chapter 16 is focused on receptors. And receptors are always proteins, and they're a receptor for something. They have a ligand. And so we've already met a receptor. It's the estrogen receptor. And we met it, and therefore met the superfamily nuclear receptors. And they're in chapter 16. Okay? So it's going to talk in chapter 16 about everything lots of stuff we just learned. That's where the figures are for the histone acetylase and the deacetylase enzymes. And I just put them out of chapter 16. And so they're down here. They're up here because they're near the start of the chapter, nuclear receptors, and we have one example of that. And then what the book does is it organizes the receptors into groups based on that nuclear receptor idea. Let's have a group, a family called nuclear receptors, and we'll pick one and teach the students about that one example. So our favorite one by far is this group called the receptor tyrosine kinases. And we learn about them in the video. The ligand for one of these is called PDGF, and that's the focus in the video. That's what's released under a scab. It is stimulating the skin cells to grow and divide. And so it's called the PDGF receptor. The insulin receptor, famous insulin receptor, fits in there, and as do things that regulate that FOXO that we started our course with. So this is where we finally catch up with FOXO. And so we'll spend most of our time talking about that family, and that's the family that has the RAS signaling pathway in it, and lots of other oncogenes are in that pathway. And that's a big reason why we'll talk about it a lot. It's super important for, you know, health and disease and stuff like that, not related to cancer. And then there's another big category that we won't talk about very much, but is a huge category of receptors called G protein coupled receptors. And we'll talk about a couple examples of that, so we can just use those examples as general examples. Then there are these, and you might call them sort of like orphan receptors. They don't fit in big categories, they're each kind of distinct. And the book has those, and we'll talk about those as well. Definitely for our favorite receptor, it will be a receptor tyrosine kinase. Even more favorite than the nuclear receptors. The next thing to say about this chapter is this concept, and that is, when we talked about the nuclear receptors are transcription factors themselves. What's going on with that is estrogen can actually float it through the blood and float right through cell membranes right into cells. The estrogen hormone and testosterone are both lipophilic molecules. They aren't very hydrophilic. And so there aren't very many molecules like them that can actually be in water and oil. One way to think about estrogen is like this. If you took, you know, just a jar with water and cooking oil in it, right? And you shake it up and you let it sit, the phases separate. And we have the hydrophilic aqueous phase and the hydrophobic organic lipid phase, like that. If you take molecules that dissolve in them and test where do they go, this is what happens. So if you take some salt and dissolve it in that mixture and shake it up and then let the phases separate, the salt, they say, partitions 100% in the water phase. There is no salt in the cooking oil phase. That's hydrophobic, and the salt is very hydrophilic because of the positive and negative charges, like that. And so there are lots of molecules that will do that. They'll partition 100% into one or the other phases. If you do that with estrogen, you get a really surprising result. Estrogen partitions not cleanly into either one. Most of the estrogen will be in the salad oil. It's very hydrophobic, but it does have some hydrophilic characteristics, and there'll be a little bit of it in the water. And so nuclear receptors are like that. Their ligands can actually enter the cell, or they're already in the cell. There are lots of different kinds of nuclear receptors, okay? And that's unusual. And so the nuclear receptors are in a different category than all the other receptors. All the other receptors are in the cell membrane, and they are membrane-spanning transmembrane receptors. Where outside of the cell, they have a binding domain for the ligand. And then they have a transmembrane domain, and then they have an intracellular domain. Because most of the ligands for this cell signaling don't come into the cell. They stay outside of the cell, and they just bind to that cell surface receptor, and they activate the receptor, and the receptor communicates the presence of the ligand into the cell. So that's what we're going to do, is we're going to talk about the nuclear receptor, and that's a great example. Then we talk about these, and that immediately brings up an issue, which is a lot of these receptors that are in the cell membrane change gene expression. And so they're going to biochemically communicate into the cell and change a transcription factor. So we saw that with FOXO on the first day of our course. There's a growth hormone ligand that binds the receptor in the cell membrane. The growth hormone stays outside of the cell, but it activates the receptor, and through a signal transduction pathway, the receptor gets phosphorylated. So we're going to have a cellular response, and it can include changing gene expression, and it can include that by targeting these transcription factors. And so it's a really interesting thing to think about, where the nuclear receptors have this direct connection between the hormone and gene expression. The receptor is a transcription factor. Then these others have to have an indirect connection. The hormone or ligand, the signaling molecule, is outside the cell, and we need to communicate its presence into the cell by having a receptor in the cell membrane that binds the ligand and then activates a response inside the cell. So that's what this chapter is all about. It starts with the receptors, but then we're interested in those signaling pathways that are inside the cell, and then we're interested in the cellular response. Our favorite cellular response is going to be stimulating the cell cycle. So you learned about the cell cycle in IntroBio, G1, S, G2M. That's chapter 17. And that's there because cancer is usually the cell cycle out of control. And that's going to be our favorite cellular response. And in this RAS signaling pathway, you'll see we pick on it for a bunch of reasons, and one of them is because there's just a direct pathway from a growth factor under your scab, and the growth factor is called PDGF, to the PDGF receptor, to RAS, and RAS is inside the skin cell, and activation of a transcription factor called ALK. And then ALK activates gene expression, and it includes a target gene called MYC. And MYC is a oncogene that was first discovered as an oncogenic retroviral gene, but it's just there's a ortholog of MYC in certainly all vertebrates. Yeah, and so that's what we're doing. You can draw that out, right? So it's a cell, and you can spread it out, and that's the assignment for the video. Lots of stuff gets talked about in the video assignment. The assignment that you do is just draw a big picture of the signaling pathway and label it, and it's a figure in your chapter 16. The book talks about it too. So that's where we're at. And again, you don't need to scramble to write these down right now, because we'll go through this and show it again and again. That's our guide through this, is to use the receptors to guide us. I tipped this on a side because this is a chapter from the previous edition, and I just didn't have time before class I recognized that, I got to change that one. But what this is just showing is just showing the table of contents for chapter 16. And it talks about the estrogen receptor, it talks about the PDGF receptor, like that. So we're not going to go through the chapter in exact order, because we're really going to get into the PDGF receptor and receptor tyrosine kinases a lot. So here's our biggest, this is going to be the ultimate big picture of our course. And so that's why this chapter is so big. Oh, and so I forgot to update these chapters. But our course is, but I'll get them updated and show you. So this is the biggest picture. Regulation of gene expression, especially regulation of transcription, that's chapter 7. And so we started our course with that. That's the MyoD stuff, is regulation of transcription. So we love that. Now, in chapter 16, regulation of cell signaling, and we especially love how that signaling can affect transcription and new gene expression. And what we really like to see is when we do things like we activate expression of a gene like MYC, because MYC can just directly drive the cells through the cell cycle. So that's chapter 17. And again, I apologize that I didn't update these chapters for this edition. This is the first year we're using this edition. Yep. So our assignment on the PDGF receptor, that video that you'll be watching, that's the center of our course. We have to learn a lot of stuff around it, but that's the hardcore center of our course, because it laces this together. And in doing so, it'll be telling us a story of our normal health, how our cells respond to signaling factors to grow, and how that can get disrupted in cancer. Yep. All right. So just want to show you this. This was the assignment we had, drawing assignment. This is the first drawing in it. You know, when we were talking about retroviruses, and there are two reasons we want to talk about it. And one is in their replication cycle, they use this reverse transcriptase. So we learned something about that and how that's a really interesting enzyme and it can be used in experiments. And then the other is that the retroviruses have to insert into the host chromosomes, and they insert in random locations. And one time, in one rat, there was one retrovirus that inserted near the RAS gene, and it accidentally copied the RAS gene and took the RAS gene with it to infect other cells. And that was a big problem because that RAS gene got defective and killed all the cells it ever infected, like that. Yep. So good assignment, you know, blah, blah, blah. This is just some drawings that one of the students did. Great. You know, very physical and these oncogenic retroviruses, of which there were about 20, each one picked up a different host gene. So there's a host gene called SARC and RAS, and MEC was one of them. And there's going to be a problem with that host gene. That's going to somehow cause out of control growth when it infects cells. So here is the RAS signaling pathway. So this could easily be PDGF, the PDGF receptor, and your actual homework assignment that's due next Tuesday is to draw this. Because we want to learn about RAS and how RAS is on this signal transduction pathway. So the signal transduction pathways are inside the cell, in the cytoplasm, like that. And RAS is got a covalently bonded lipid molecule. So RAS is tethered to the cell membrane. It floats around in the membrane side to side. It's on the inner surface, and it's stuck there because that's where it does its job. And so this is just a click, click, click pathway for the response when that growth factor is present. And our favorite growth factor is PDGF. It's released from platelets, so it's called platelet-derived growth factor, PDGF. And it gets released by platelets, most famously under every scab, and it stimulates the skin cells. So if that was PDGF and a PDGF receptor, this could be a skin cell under the scab, getting stimulated to go through the cell cycle. And we'll learn in chapter 17 in the cell cycle chapter that the skin cells are constantly going through the cell cycle, right? We famously have dead skin cells rubbing off every day of our life. So the skin cells are going through the cell cycle slowly, and they're going through it slowly because there's a place where they wait. And the majority of the skin cells in our bodies right now are just waiting, and every once in a while they get to go. So we get a slow replacement of skin cells. Under a scab, we just want to speed it up. What's the rate limiting step? The rate limiting step in skin growth is the point in the cell cycle where they wait. And so the target is going to be to get them through that point in the cell cycle more quickly. And they'll grow faster and heal. And you know, when the scab falls off and you have all that pink skin under there, lots of new skin cells. Yeah, it's as simple as that. And the pathway is really simple. And that's one reason why there are some really famous oncogenes on this pathway. Because it's vulnerable. It's kind of like an Achilles heel. There's lots of checks and balances, but it's a little bit dangerous to have it be set up the way it is. And so RAS is there, and RAS can be mutated in a third, I think, about a third of all human cancers. It's contributing. We'll learn as we get into cancer that it usually takes multiple different mutations for a cancer to really grow out of control. There are checks and balances and so forth. And but RAS is one that's really commonly mutated. And there's a reason why, and we'll learn maybe today about why RAS is so vulnerable to cause, to get mutated and cause cancer. Yep. So anybody have any questions about that? And so, you know, the book puts us in two figures. It's a little bit frustrating, right? That you got to go, you know, you're in the cytoplasm. You got to get into the nucleus, and you got to phosphorylate ELK. And so ELK is the one in transcription factor we already talked about. It has to get activated to, and to get activated, it has to get phosphorylated.
Dave, the G protein exchange factor.
Yeah.
That starts off the RAS changing to a GTP.
Yeah.
Has an SH2 domain, an SRC homology 2
Yeah.
domain. Is that the same domain that we're going to find on the non-catalytic tyrosine kinase receptors?
No, I don't think so. So this is just this gets into the weeds some, but when the scientists in the 1970s were studying these oncogenic retroviruses, they found about 20 different genes. And I mentioned one is called SARC, like that, okay? One is called RAS, and one is called MYC, okay? So let's just put those on those names on this list. So there's RAS, and RAS is on a normal growth stimulation pathway, and the way it works is the RAS is going to get activated, and it's going to stimulate growth. So when RAS is activated, it's going to stimulate growth. So RAS is there. Then way down here at the end, one of the target genes that ELK is going to activate is called MYC, M-Y-C. And so that's another oncogene. And MYC can just directly stimulate the cell to go through the cell cycle. It can activate, MYC is itself a transcription factor, so we'll get to MYC because it's a transcription factor. And it's got target genes that scientists have identified using CHIP. And it activates cell cycle factors, okay? So we have, along that pathway, proteins we're interested in. So Sam asked this question about this. And so this C is called an SH2 domain. And you can see that it looks like it's kind of building a bridge on this pathway, right? We're making connections on this pathway. It's like, if you know that there's an old board game called Mousetrap. And Mousetrap has its thing where it's like it has different steps and it just kind of kicks through them, kind of like dominoes falling, but they're all different, kind of different things. And so different things are happening here, like click, 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 click. And so we'll just go through them to introduce them. So when the PDGF binds to the PDGF receptor, it actually helps the two monomers of the PDGF receptor stay together. And receptor tyrosine kinases are all in the same group because the receptor is a tyrosine kinase. And a kinase phosphorylates targets. And it can, if it phosphorylates proteins, there are only 3 amino acids that can phosphorylate, tyrosine, serine, and threonine. And serine and threonine kinases are very common, tyrosine kinase is less common. So it's called a receptor tyrosine kinase, because that's what it does. It's a receptor that has a tyrosine kinase activity. And the kinase activity is surprising to see. When the two monomers of the receptor, PDGF receptor, get held together, this monomer phosphorylates that monomer. So these phosphates were added by that one, and those phosphates were added by that one. And now the receptor is active. When it's phosphorylated like that, on the inside it's active. So see, the receptor works kind of like a teeter-totter, where when one end changes, the other end changes. A teeter-totter or a pair of pliers, there's a conformational change, a difference in the situation. The growth factor helps so that these are close together, and that tyrosine kinase activity phosphorylated, and now that's active. As long as that's phosphorylated, it's active. And so that's the next step, is to have a protein bind to them and take that activity on to the next step. And that's what's happening here. And this is shown in 2 colors here. It's really just one protein. And this one protein has a binding domain that binds to the phosphorylated receptor and activates it. And then that activates the RAS. So now we're bridging from the phosphorylated receptor, active form, to the RAS. Let's activate RAS. And it's just really one protein. Why are they doing it in 2 colors? Why are they emphasizing that? Because one of the, probably the, back in the 1970s, the most famous oncogene that they found in an oncogenic retrovirus they called SARC. And this is a relative of SARC. It's got a homologous region. It's not an ortholog or a paralog. It's only kind of related, kind of similar to SARC. And that's what SARC is like. SARC is not as easy to point to as RAS or MYC. It's one of these really famous oncogenes that's very important in cancer, and it was discovered, 60 years ago, but it's just hard to point to it. So that's what they're doing there, is they're just pointing it out. So it's kind of a confusing point out, but it fits on this pathway. It could be mutated, and you could get the pathway going without a growth factor by, accident. Yep. So that's where that fits in. Yeah. And not, it's kind of a confusing thing, and we have really elegant examples of other oncogenes and their action. Like, let's talk about RAS, just to dive into this. So why is RAS so often found in cancers? And there's an answer, and the answer is that when you find RAS in cancer and it's mutated, the mutations are always in the same place, in the gene. And so let's draw the RAS gene and talk about where the mutations are, right? Oh, there we go. All right, so get this switched up here. So, yep, oh, no, gotta do that too. So there's a RAS gene. And, it's got a regulatory region and so forth like that, right? So we know all about that. I don't have to label it. You could label it. You could label it in your sleep, hopefully. And I don't have to, okay? So RAS is a coding gene. It codes for a protein. It codes for the RAS protein. Then there must be a coding region here. And when we look at cancer tumors that have a mutated RAS, it's always mutated in the same place. Never over here. For RAS, it's never a regulatory story about, switching the gene on. The mutations are always in the coding region. Always. And there's only a couple of amino acids that ever get mutated. So it's just changing one amino acid, and there are just a couple of them that are ever mutated. And we can look where. So let's look in this protein, and here's RAS protein. And it's an enzyme. So there's the active site of the enzyme. And the mutations are always in the active site. There's been an amino acid change in the active site, and so the activity is decreased in cancer. All right? Always those mutations. And we'll go back over this because it's just crazy to see this, in understanding that, how it works. Now, why does this happen so often in RAS? Okay, well, here's the deal. These mutations are hypomorphic or loss of function mutations. They're a decrease in activity. We still have RAS, but that enzyme activity that it has decreased. And those are the most likely mutations. Mutations are random. And so they're most likely to decrease the activity of a gene, not increase it. Yeah? And so it's really interesting to say that the mutations in RAS, when you see it in cancer, are decreasing its activity, but increasing the cell cycle. So what's the deal there? And so it's just a little surprise, but it explains in a really clear way why RAS is so common in cancer. First, it's a normal protein that's normally involved in lots of growth control. So it's there in a lot of cells. Second, random mutations that decrease its activity increase the cell cycle, right? So, and your book talks about this too, but let's just keep rolling on this because we dive into this a little bit, dive into this topic a little bit, okay? Okay, so here's how RAS works. So we can draw it like this, and now we have to draw over here probably, I don't know, maybe I don't want to draw it that clearly because it's not that clear. So let's just draw a RAS here and say that's RAS protein. And we're really interested in the fact that it's an enzyme and it's got a catalytic activity, okay? So then the next thing to say is there's a name for RAS, and the type of protein it is, it's called a monomeric G protein. And that's the only monomeric G protein we're going to learn about later in this chapter, just to say it and not confuse you, but let you see that it's very different. Later we're going to learn about some other G protein that's called a trimeric G protein. So that's why we're calling it monomeric, is because it's a very different story than the trimeric G protein story we'll learn about later. You know, so not the world's biggest deal, but it's definitely a G protein, and it's definitely a monomeric G protein. Okay, so what is a G protein? And a G protein is it always has bound to it either GDP, and when it has GDP, it's inactive, or GTP, and when it's bound to GTP, it's active. So that's what it's meant to be a G protein. It always has GDP or GTP. And GDP and GTP are like guanine triphosphate or guanine dinucleotide triphosphate. And so that's the same idea as ATP. You're more familiar with ATP. So you can have ADP or ATP. And the difference is just the phosphate. So it's like that. And it's just the G of the nucleotide AGCT. Example is just the G. And so we could draw on here, and let's just do it like this, where we say that we start out with an inactive inactive RAS, and then there's a wound, and so this PDGF binds and activates the PDGF receptor. So PDGF coming outside the cell, and then here's the PDGF receptor binding to it. So a receptor tyrosine kinase gets activated, and that leads to the activation of RAS. And so before there's PDGF and it's inactive, this RAS has a GDP bound. It's got a GDP bound. Oops, not very well written, but GDP, inactive. And we're going to activate it, and when we activate it, we're going to exchange. The activation involves just exchanging, so it's going to let go of GDP, and it's going to replace it with GTP. So it's an exchange. So now it's active, and we draw GTP here. That, and it's active. So see, the way it got activated was an exchange of molecules. There was a GDP, and it let go of that one and grabbed hold of GTP. So an exchange of molecules. This RAS is going to be active until its enzymatic activity hydrolyzes GTP back to the GDP. And that's that enzymatic activity, and now you can see what happens. So in this case, the enzymatic activity is to turn it off. And if we break it with mutations, it won't turn off. So that's kind of like the weak link there, is that random mutations are likely to break the protein, and this protein needs to turn itself off. And if it's broken, it doesn't turn itself off, and it just keeps going. You go, 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 go. It stays active too long. You know, we want to give a quick stimulation, go, skin cells, go, and then stop going. We don't want to just keep go, go, go. So when RAS is mutated, even without the growth factor, it's saying go, go, go. So even in a person's skin cells that are not wounded, it's active, and it just can't turn itself off. Yep. And so, you know, and every time you talk about the molecular mechanism of cancer, you run into examples like that, where you're like, oof, dang it. You know, that's the problem, is it just can't turn itself off very well. You know, and that's a really vulnerable thing, right? Because it's pretty easy to damage a gene so that the product can't, you know, function. And that's a function of it, is to turn yourself off. The book, it's a little bit confusing because the book mentions that there is actually another protein that also bound to RAS that actually helps stimulate that activity. So, you know, when you get into sort of more advanced details, there's more added. But we can just disregard that other protein because it's the RAS that has the enzyme activity, and the enzyme activity, to be official about it, we can say is GTP hydrolysis. So RAS, and you can draw RAS here as an enzyme in a chemical reaction where we're going to catalyze the breakdown of GTP to GDP and inorganic phosphate. Yep, that's the reaction. And it turns itself off because GDP is the inactive form. So remember, to activate RAS, we actually exchanged molecules. We let go of a GDP and we added a new GTP. To turn off RAS, we don't let go of the GTP, we just hydrolyze it. Yep? Okay? And so, and that just, you know, dove into a little bit to address that question about, yeah, see, RAS is fascinating and significant. Fascinating for learning, because you're like, oh, I get that, and that's a, you know, it's this clear message. And back to Sam's question about the Sark homology domain, that's just more kind of, it's harder to kind of explain, and it doesn't bring such a clarifying, you know, like nobody's going out and getting tattoos of that Sark homology domain, because they're just like, yeah, it's really a big deal, but it's just not really available for good, memorable learning. Yep? Yep, okay? So people have questions about that. So why don't we just do a concept chat earlier in our class meeting today instead of later, just because we've run them late and we had to get out of here. Because we'll do it, then we'll keep talking, okay? So let's find a partner, and again, try to find two, find a group of three, try to split it up, and somebody new, each time. And make some friends. You can say something about, the stuff you're interested in, things and stuff like that. But we'll do this for a couple minutes, and then we will check in. Okay, so we'll do some concept chat. What are three important things you think we already talked about today?
You're saying that it's mutating into a state where
yeah, it's the actual like enzymatic function.
Oh, yeah, because it's always the active site. Okay, so it won't let this bind to the active site of the enzyme breakdown.
That's a good question. I don't know if it's binding to the active
Oh,
site.
it might not be the active site though.
Well, or it could be other components of the active site.
That's weird. Oh, that's like, you know, it's like most enzymes, especially these ones in tablet systems that have like a binding factor on the back, which is where the ingredients would be, which if you change the active site, it will change the binding site.
Sure. But I think it could be like it's still
And we'll just do it for another minute.
right, but then it doesn't have a nucleophile. Or maybe it now like yeah, so or it could be the binding or it could be that.
Oh, yeah, and then we'll check in. Cameron, what is one thing your group talked about? Yep.
Oh, we were talking about the chemical part of GTP getting hydrolyzed into GTP and phosphate.
Yep.
So
Yeah.
the question that I had was it the active site changing the catalysis of GTP to GDP and PI, or was it the site where it was bound on the back and the active site changing?
Yeah, that's a good question. And I kind of stumbled, when I said, I want to show you, RAS protein with GDP or GTP bound. Because it's a little bit complicated because, usually when you think of an enzyme, you think, oh, it's just going to grab it and it's just going to catalyze a reaction. And the binding site, the G binding site on RAS is in the active site. So it's a little bit of a complicated active site where it is always bound and it wants to, if it's GTP, it wants to break it down to GDP, but it doesn't immediately do that. You could think of it as that its natural catalytic activity is pretty slow. Yeah? And so, again, with enzymes, we're used to thinking about enzymes that are just going to grab the substrate and go really fast and let go of it. And in this case, the RAS is going to have the substrate on it, and it's just not going to break it down very quickly.
You said there was a cofactor for the breakdown?
Yeah, so there's another protein that binds, and I want to mention that because the book has a figure with that in it, and the book talks about the other factor as if it's really the other factor that's the doing the important job. And it's part of the story, but remember the mutations that cause trouble are in the RAS gene and not that helper protein gene, because it's the RAS gene that's got the actual active site that does the catalysis. Yep.
Well, how does it make sense that it's only the active site mutation that changes the outcome?
Because that's where what's happening is where it's regularly going pretty slow, and the mutations make it go slower. Right? We want to break it down pretty soon, but we definitely want to break it down. And we're going to use the catalytic activity to break it down. It's just going to slow. It's going to be a little bit, it's going to take a minute, like that, And I should mention that, we haven't mentioned this yet. For these receptor pathways, there are, for the cell, like the skin cell, there really aren't very many PDGF molecules that bind. So these receptors are very sensitive and responsive. And that's because there are amplification steps. So we're going to increase the response to a little bit of a signal. And that's part of the RAS story, is by having a slow breakdown, normally, it helps amplify the signal. So just one PGF molecule can get kind of a good, strong signal into the cell. So that's one reason why, the reason why, the enzyme would be slow. And then the mutations are in that active site and they slow it down even more. They slow it down too much. And then that's bad because then it stays active. It's active too much. So it stimulates too much. And we'll see that it's good to bring that up because that amplification is a problem with these pathways, right, is if we get too much amplification, we'll get too much cell cycle, we'll get too much out of control growth. Yep. And so there are lots of steps on these pathways that involve amplifying. And enzymes are good at that, right? An enzyme goes click, 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 right? And it's amplifying. It's making a lot of product with just one enzyme molecule. Yeah. So, yeah. And this was, we kind of dove into this, right? But this is something we'll loop back over a bunch. It's big in the video. I talk about the mutations of the video for our assignments, stuff like that, right? So it's just kind of a first pass, but it's something that it takes several passes. So it's great, we're concept chatting it, and then questioning, clarifying, and being like confused, and being like, it's got to sink in. That kind of learning is good, right? Because learning requires recall, and if you don't understand something, you keep thinking about it. You're like, I still don't get that, and you're thinking about it. So it's actually good to have somewhat challenging learning, because you have to think about it. Yeah, so that might help some for now, Cameron, like that, and it's starting to kind of like, okay, I'm thinking, right? And challenge it, right? Be skeptical and challenge it and come back and ask about it, because remember, this thing is mutated that way in almost a third of all human cancers. Yeah, so Yusira.
I don't know if this question makes sense, but is there ever a case where the cells like, oh, we're making too much of this, there's something wrong with the rise or whatever, and is there like a negative feedback that stops the receptor tyrosine kinase?
Yeah.
Not phosphorylate?
Yes, so let's just write that down, because, and again, we're diving into this, and so if we look at like this figure, and we draw on it, so here's the signaling molecule, and they're always small, so we could call them ligand. In some cases, they're hormones like insulin. In other cases, they're local signaling factors like PDGF. Then here's the receptor. They're almost always in the cell membrane because the ligand is not going to come into the cell. So nuclear receptors are super first examples to learn about, but they're already in there, and the ligand gets in. So this is the typical receptor. It's in the cell membrane. It's called, a transmembrane protein, because it has an extracellular domain that's the ligand binding domain, and then it has an intracellular domain that gets activated. And so there's that. And then all of this is called the signal transduction pathway. So it's happening in the cytoplasm, it's inside the cell, and there are a bunch of different ones, okay? And so there are really, the first look at a signal transduction pathway is, that's how we're getting the signal communicated into the cell. The membrane's on the cell surface, and we want to communicate the signal into the cell because we're eventually going to have a cellular response. The cell is going to do something in response to that signal. And our favorite response is going to be chapter 17. It's going to go through the cell cycle. It's going to grow and divide. Okay? All right, so there are really two stories in a general sense about these signal transduction pathways. And one we just talked about, and that is they amplify the signal. So they're involved in amplification. It just takes a little bit of the ligand to get the whole cell to respond. And if you look along here, there are several examples of receptor kinases on this pathway. And a kinase is an enzyme, and I mentioned enzymes are very good at amplifying something because one enzyme molecule can catalyze a bunch of chemical reactions and make a bunch of product. So there are several enzymes on this pathway to amplify it. And then Usura just asked about feedback and regulation, and there's tons of that because we're playing with fire here. When we're talking about growth control, that is dangerous because when it's out of control, it's cancer. So the other thing that's really important for us to learn in a general sense about signal transduction pathways is called integration. And that means that a lot of times it actually requires two different receptors to get activated to really get a response because of this feedback and crosstalk between them. next Tuesday, Sam's going to come back and he's going to talk to us the next step in that CAR T-cell therapy story. And he already told us that the scientists took 3 different receptors and they put them together to make one super receptor. And what they normally are is they're normally separate receptors, and they all need to be activated to get the T cell to respond. And what they did is they just combined all that into one receptor, one super receptor, and they just made it so there wasn't as much feedback, so much integration, so much checks and balances. You could think of that integration as being like checks and balances. Is this the right thing? Is that the right thing? And so, yeah, somebody can have a mutated RAS, but that doesn't lead by itself to cancer, right? It's a common mutation in cancer, but the person will have other mutations in cancer. There's a good chance you've heard about the idea that a cancer progresses. At first, it's not even noticeable, and then it gets worse and worse and worse because it's picking up more mutations and more problems like that along the way. Yep. So, Sarah, since we're back there, what's something else your group talked about?
We talked about how RAS activates ALK, and ALK is the transcriptional factor that leads to cell progression.
Yep, so we'll see that in the next chapter. So right now we want to start leaning into it. We want to say that activated ALK is going to activate expression of genes that are important in stimulating the cell cycle, stimulating the skin cells to grow faster, right? They're normally growing slow. They're going to grow faster. Yep. And one of the targets of ALK is this MYC. And MYC is a transcription factor that's going to help. It's going to help activate expression of genes that are required to go through the cell cycle. And it turns out that that's just so satisfying. The story of ALK and MYC, we'll talk about kind of a little bit in this chapter, and then more, because we'll start leaning into chapter 17 of the cell cycle about how it regulates it. Yep. Let's see. So great. And then Teresa. Is Teresa here? No, because I don't know Teresa yet, and I have her quiz, so I thought I'd ask about Teresa. So how about Isabella?
We talked about how the monomeric protein always has the GDP or GTP to it, and GDP is inactive, and GDP is active.
Yep. Yeah, so it's a G protein, and we're going to learn about another G protein later. So let's make sure we keep a track of it. So it's a monomeric G protein. And that just means it's just, one protein molecule makes a RAS protein. And it's a G protein because it's always bound to either GDP or GTP. And the way it gets, and it's inactive when it's GDP, and the way it gets activated is it exchanges the GDP for GTP with the help of this GDP exchange factor protein. And then it's active, GTP. And it stays active until it actually cuts GTP back down to GDP, and then it's inactive. Yep, okay. So questions people have about any of this stuff? Yep.
When you were drawing the inactive RAS protein, you drew the PDGF next to it.
The PDGF next to it?
Yeah, was there like a purpose behind that?
No, it was a mistake. I mean, it wasn't a mistake. I just drew them too close together because they're physically on the other side of the cell membrane. The PDGF is outside and the RAS protein is inside. Yep. And so just something to mention is in the book, the author likes to make these little sunbursts around some proteins. And the sunburst it's making it wants to point out proteins every once in a while, and it doesn't talk about them really, but it's pointing out proteins that can be mutated and cause cancer, can contribute to cancer. And proteins that can have their genes mutated that can help contribute to cancer are called oncogenes. And so RAS is an oncogene, MYC is an oncogene, SARC is an oncogene, RAF is an oncogene. And we'll really learn about RAS, and the rest of the proteins of this pathway much less about. So you can see that activated RAS, when it's activated, what it does is it binds to RAF and it activates RAF. And RAF is a protein kinase. RAF phosphorylates MEK, and MEK is a protein kinase, and when it's activated, it phosphorylates ERK. And when ERK is active, it's a protein kinase. Inactive, it phosphorylates ALK. And you can see those proteins, they're really important in our bodies, but they're kind of boring because they're all doing the same thing. And what are they doing? They're amplifying the signal. They're all enzymes, kinases that can activate a bunch, right? So one activated RAF can activate a bunch of MEK molecules. One activated MEK can activate a bunch of ERK, like that. So RAF, MEK, ERK, ALK, not the world's biggest deal, but you know, that's part of the pathway. You know, not the world's biggest deal, not compared to RAS or MYC. And MYC isn't on here, but it's down there for the target genes. Yep. So other questions people have about this? And this is the showcase receptor and signaling pathway in the homework assignment, the PDGF receptor and the skin healing under a scab. Then it's also a showcase receptor in our book as one of the members of the receptor tyrosine kinase group. Like that, they all fit together into the same pathway. Some of the receptor tyrosine kinases use this signal transduction pathways. Some of you use different ones. And we're going to learn 3 signal transduction pathways. The RAS one and 2 others from this category of receptors. Just to start to show you that there's a bunch of variety in the pathways because they have to lead to a bunch of different types of cell responses. We'll focus on stimulating the cell cycle, but there's lots of different things that cells can do. Sam's going to talk about that on Tuesday, right? The T cell responds by stimulating the cell cycle, but it also is going to respond and kill the cancer cell. So it's going to be, you know, several responses. And so we want to learn some about that in that chapter 2. So we'll spend more time talking about the receptor tyrosine kinases than any of the others. Okay? All right? Doing great. I think we have another minute or so. So let's just see. Yeah, we have 5 minutes here. So that's where we're at. And so that's a figure I used from Intro Bio textbook because remember that this is really first semester Intro Bio. That's what this course is. And why was it taught in first semester Intro Bio? Because it's just the most significant biology. It's just amazingly consequential. And so let's get it out there. But it's really hard to learn Intro Bio in Intro Bio and have it stick and really like I'm walking around and could, you know, explain what's going on there. Yep. So we're going to learn over time probably about a half a dozen of the famous oncogenic retroviral oncogenes that were discovered in the 1970s. And we'll talk about SARC, but not for the first exam and not with any great significance. And so we'll learn about, for our first exam, MYC and RAS, and we'll learn about these others for the second exam, because for our first exam, we'll probably get through this chapter and then the cell cycle chapter, and then we'll have an exam. And then the next chapter is about programmed cell death, and the next chapter of that is about cancer. And so these other oncogenes come up in that, especially in that last chapter in our book, chapter 19. So we're doing great, and just remember that these popped up, and they're important in cancer. You know, they're out there in cancer, and but scientists have been studying these the longest because of how they popped up in the 1970s as getting our like foot in the door for the molecular mechanisms of cancer. So we'll pause here, and you know, again, I'll probably send you these, share a Google Doc with you that we'll talk about on Thursday to get us rolling on our term paper stuff. All right?